Hello, my name is Sebastian Springer and I'm a professor of biochemistry and cell biology at Jakobs University Bremen in Germany.
And hello, my name is Susanne and I'm a former PhD student with Sebastian.
So we thought today we would tell you a little bit about our new paper that just appeared in the journal Traffic. It's very close to our hearts, something we've thought about for a long time, and now we'd like to tell you about it. So the main question is, how do different proteins travel from the ER to the cell surface at such different rates or speeds? Why is that so?
Yeah, and to address this question from a very general point of view, we studied a model system of two very similar MHC class 1 proteins of the immune system.
MHC class 1 proteins are type 1 transmembrane complexes. They contain a relatively large luminal domain, a short transmembrane segment, and a cytosolic tail. MHC class 1 proteins are trimeric, for they have the heavy chain, shown in light brown, a soluble light chain, called beta-2 microglobulin, shown in pink, and a small peptide fragment that binds to the top of the molecule. In this presentation, MHC class 1 proteins will be represented by the symbol that you can see on the right. The peptide is represented by a green circle, and in pink you will see the beta-2M subunit. MHC class 1 proteins are assembled inside the cell in the endoplasmic reticulum, or ER, with the help of multiple ER chaperones, for example calnexin, tapazin, or reticulum. During the assembly process, the heavy chain binds to the beta-2M subunit and the peptide, and only then the fully assembled trimeric complex will traffic in the ER to the ER exit site and then follow the secretory pathway to the cell surface. For MHC class 1 proteins, many different allotypes exist. Some of these differ only in a few amino acids. For us, H2DB, shown in orange, and H2KB, shown in blue, differ in about 20% of their primary amino acid sequence. As a result, H2KB traffics from the ER to the cell surface much faster than H2DB.
And to figure out why the trafficking of DB and KB from the ER to the cell surface is so different, we analyzed the transport in terms of ER exit, ER to Golgi transport, protein flexibility, protein stability, and for this we used theoretical and experimental approaches.
So what we found out is that each protein in the early secretory pathway has a certain affinity, a certain binding affinity to all the other proteins that are also in the early secretory pathway, for example chaperones and other factors. And this affinity of a protein to these other proteins depends on its folding state and its maturation state, perhaps its glycosylation, and so on. And it is that affinity of a protein to the other proteins of what we call the matrix of the secretory pathway that determines the rate of transport of that protein from the ER to the cell surface. And that, of course, differs from each protein to the other. And so now let us tell you how we came to that conclusion.
In an in vitro COP2 budding assay, we demonstrated that H2DB and H2KB traffic differently to the cell surface because of a step that takes place inside the ER and before the arrival at the ER exit sites, so here. In this in vitro COP2 budding assay, we generate these ER to Golgi transport vesicles, or COP2 vesicles, in vitro from isolated ER membranes. We then compare the amounts of H2DB and H2KB inside these COP2 vesicles. We found that H2KB, directly after synthesis, is present much earlier in these COP2 vesicles than H2DB. So prompted by this, we went ahead and studied domain exchange mutations of H2DB and H2KB. For this, we transplanted luminal, transmembrane, and cytosolic domains onto the other molecule. For example, we compared the wild-type H2KB molecule on the left to a protein that consists of the luminal domain of H2DB and the transmembrane and cytosolic domain of H2KB on the right. From these kind of data, we found that the luminal domain contributes mainly to the trafficking phenotype of H2DB and KB. So all the constructs that carried the luminal domain of H2DB were significantly slower than their H2KB counterparts. In other experiments, we could present further evidence that this is not due to the foldedness of the luminal domains of H2DB and KB, so they are folded to the same degree. But interesting, we found in molecular dynamic simulations that the luminal domain of H2DB is much more flexible than H2KB. And based on these and other results, we propose that the cell surface transport rate of MHC class I proteins and probably many other type I transmembrane proteins is governed by the affinity of these proteins to the ER matrix, so carnexin, tepezin, and reticulum.
Yeah, so of course the detailed experiments are all in the paper, and so we hope that you will read that, and we hope that we've convinced you this is interesting, and also that MHC class I molecules are a fantastic model system to study protein trafficking in the secretory pathway.
So, thank you for watching this video, and if you like our work, get in touch with us.